At medical centers across the country, researchers are steadily uncovering clues to the mysteries of interferon. While the big push is to determine interferon's cancer-fighting potentials, some scientists are applying interferon research to other areas of medicine. As if almost by chance, two areas of investigation have come together here at the University of California in San Francisco, with surprising results. Dr. Lois Epstein is an immunologist studying the body's natural defenses against infection. For several years now, she's been exploring interferon's complex relationship to the immune system, how certain cells respond to interferon, and how it might be used as an anti-tumor agent.
We now have systems in the laboratory whereby we can actually take tumor cells from individual patients and study whether those cells are sensitive to the anti-proliferative effects of interferon.
These three photographs, taken at 7-day intervals, show the first 3 weeks of life of a test tube tumor. In over 70% of the samples tested, the tumor shrank with the presence of interferon.
A great deal of work has to be done to determine first which tumors are sensitive to interferon, secondly if the anti-tumor effects of interferon are permanent and lasting, and whether in fact interferon is any better than any other standard agents that we have for treating tumors. In other words, we're trying to get at the basic biology of interferon so that we will amass information that might be able to tell clinicians which tumors might be sensitive and how does it act, because this would influence the way that they would give the interferon to the patients.
To improve clinical use of interferon, one big question still needs to be answered in the lab. What is it exactly that determines a cell's response to interferon? Well, part of the answer has come unexpectedly from recent work in pediatric genetics, headed by Dr. Charles Epstein, Lois's husband.
My involvement with interferon actually started with an interest in an entirely different problem, which is the problem of why extra chromosomes cause problems in development and problems in functioning. In specific, we've been very interested in Down syndrome, which is a human disorder which results from the presence of an extra chromosome, the chromosome which we call number 21. So rather than having 2 of those, individual with Down syndrome has 3.
Children with trisomy 21, Down syndrome, are known to have severe immunological problems. They're unusually prone to infection and have a higher than average chance of developing leukemia. But no one really knew why. Conferring with his favorite immunologist, Charles began to suspect some sort of biochemical link between Down syndrome and interferon.
It turned out purely by chance that one of the genes which has been found to be present on human chromosome 21, a gene which was identified by a certain type of mapping techniques which are used in genetic research, that gene turned out to be what we think is the receptor for interferon. It's the gene which controls the production of what the cell needs in order to see interferon so that it knows it's there.
When trisomy 21 cells were put into culture and then treated with interferon, the response was 5 to 10 times greater than in normal cells. The Epsteins attribute this increased sensitivity to the fact that with the extra chromosome, there is indeed an extra dose of interferon receptor. And it's this heightened sensitivity that may hinder the immune system's ability to fight off infection.
To study such problems in humans is very hard. To study any developmental problem in humans is very difficult. So as a way of trying to get beyond that block, we have recently turned our attention to seeing whether we can develop in the mouse a model for at least some aspects of trisomy 21 or Down syndrome.
Now, humans with Down syndrome can survive relatively normal lifespans. Unfortunately, mice with the same affliction almost always die before birth. So, taking nature by the hand, the Epsteins set out to breed mice that would develop a syndrome resembling the human disease.
We do that in the form of making what we call chimeras. an animal which actually starts from two different embryos. The embryos are combined and you end up with an adult animal that has two entirely different sets of cells, a normal set of cells and a trisomic set of cells. A reasonable model for certain aspects of human trisomy 21, but it'll allow us to study some of the functional issues that we're interested in getting at with regard to what interferon may be doing to cells which have an extra dose of the interferon receptor.
If we deal with cells that are more sensitive to interferon, we have excellent systems for trying to understand better the mechanism of action of interferon itself.
If one has some model system that mimics some of the difficulties that the human has, then one has an experimental tool for looking at ways of trying to alter the situation, to improve immune function, to do whatever it is that you might be able to do to change what it is that causes the abnormalities in human. You can't change the physical abnormalities. Potentially you could change some of the functional problems.
More information has to be accrued by various investigators all over the country and the world about the basic biology of the substance. And now that we know that there are so many different types of interferons, each of which might have a specific range of action which is different from the other, we have a lot more to learn before the final word is in.
Like detectives in a great mystery novel, Charles and Lois Epstein are probing around in the basic elements of biology, looking for important clues that could unlock the secrets of interferon and genetic dysfunction. Until more facts are in, however, no one knows how the story will end. But then, in research, as in a good mystery, the only thing one should expect is the unexpected.